Hello children, welcome to biology class. We are discussing the lesson Cell, the unit of life. Today we are going to explain about nucleus. Nucleus is considered as the brain of the cell. Children, nucleus as a cell organelle was first described by Robert Brown in 1831. But later, the material of the nucleus, chained by the basic dye, was given by the name chromatin by Fleming. Children, the interphase nucleus has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Nucleoli, singular form as nucleolus. Okay, so this electron microscopy has revealed that the nuclear envelope, you can see nuclear envelope here. The nuclear envelope, which consists of two parallel membranes with space between about 10 to 50 nanometer size, called the perinuclear space. So you can see space here, perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Okay. So the outer membrane usually remains continuous with the endoplasmic reticulum. You can see it in this structure here. You can see endoplasmic reticulum and also bears ribosomes on it. Okay, so also bears ribosomes on it. Then at a number of places, the nuclear envelope is interrupted by minute pores. Can you see nuclear pores here? Minute pores which are formed by the fusion of its two membranes. Okay. So what is the function of this nuclear pores? The function of nuclear pores. These nuclear pores are the passage through which movement of RNA and protein molecules take place in both directions between nucleus and the cytoplasm. Okay, so normally there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Some mature cells even lack nucleus, example erythrocytes of many mammals and sieve tube cells of vascular plants. Okay, then about the nucleoplasm. Here you can see nucleoplasm. It is a nuclear matrix or nucleoplasm contains nucleolus. Can you see the nucleolus here, children? Nucleolus and a thread-like chromatin. The nucleoli, the content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. And its function is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. So in higher classes you will study in detail about protein synthesis. Again you can see nuclear pore here, then thread-like chromatin, then nucleolus and the nuclear matrix, nucleoplasm and outer covering nuclear envelope. I hope the structure is clear, isn't it? Children, you may recall that the interphase nucleus has a loose and distinct network of nucleoprotein fibers. See, that is chromatin, isn't it? But during different stages of cell division, cells show structured chromosomes in place, isn't it? See, here you can see chromosomes instead of thread-like chromatin. You can see chromosomes here. So as a result of nuclear division, chromosomes will be present in the place of chromatin. So the chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. So we will study in detail about histone next year, but keep it in mind, chromatin contains DNA and some basic proteins called histones, then some non-histone proteins and RNA. Okay. Children, a single human cell has approximately 2 meter long thread. See, elongated structure, 2 meter long elongated structure, long thread of DNA distributed among its 46 chromosomes. Being Homo sapien, we know we are having 23 pairs of chromosomes, isn't it? In total 46. So we'll study in detail in next class. That means in class 12. Okay. So now let's see 2 meters long. Its length is 2 meter long approximately. Clear? Then now let's discuss the shapes of chromosomes. Every chromosome essentially has a primary constriction which is known as centromere on the side of the disc-shaped structure called kinetochores. Okay, so these are kinetochores. See, dark color, you can see kinetochores. This is centromere. Then based on the position of centromere, the chromosomes can be classified into 4 types. Which are they? Metacentric, submetacentric, acrocentric and telocentric. First of all, let's start our discussion with metacentric chromosome. It has a middle centromere. So we are dividing this chromosome on the basis of position of centromere, isn't it? So see, where is the position of centromere in metacentric chromosome? Yes, on middle, isn't it? So the metacentric chromosome has middle centromere forming 2 equal arms. So you can see this is P arm, this is Q arm, but both the arms are equal in size. These type of chromosomes are called, yes, metacentric chromosome. Clear? Next one, submetacentric chromosome. What is the speciality? Where is the position of centromere, children? In submetacentric chromosome has centromere slightly away from the middle, isn't it? It is not in the middle, not in the accurate center, but slightly away from the middle. These categories of enzymes are called submetacentric. Okay, and it resulting into 1 shorter arm and 1 longer arm. Here you can see 1 pair of shorter arm and longer arms. Clear? Then third one, acrocentric. In case of acrocentric chromosomes, the centromere is situated close to its center, forming 1 extremely short arm and 1 long arm. That is acrocentric chromosome. And the last one, telocentric, here these chromosomes has a terminal centromere. Clear? So sometimes a few chromosomes have non-staining secondary constrictions at the constant locations. This gives the appearance of small fragments called satellite. See, this is a diagram of satellite chromosome. Here you can see the secondary constrictions. Clear? Secondary constriction. Okay, so here centromere and kinetochore are clear. Okay, so this you can call it as primary constriction, but only in all the type of chromosomes only primary constrictions are there, but only in satellite chromosome you can see a secondary constriction. Clear? And the microbodies, children, many membrane bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cell. Okay. So that's about the lesson cell. You have to read the textbook thoroughly, children. Okay, read the textbook and study the structure and function of prokaryotic cell and eukaryotic cell. Okay, so this lesson is very important, and next class, that is in class 12 also, we are studying about the molecular biology and genetics. In all the lessons we need a basic idea about the structure of cell. Okay, so that's why you have to study thoroughly. Okay children, thank you.